Well, it's my pleasure today to tell you about what we've learned using brain imaging technologies to identify the circuits that are functioning abnormally in mood disorders and what those findings teach us about paths for novel treatment development in bipolar disorder. Among the sources of research support, as Bob mentioned, BBRF through NARSAD grants has been one of the really crucial means of support for me. And then after a couple of decades in academia, I joined my friend and longtime collaborator from NIMH, Husseini Manji, about 2 years ago at Janssen. And both Husseini and I had thought that, that the field had enough new leads now that it was really time to develop new treatments for mood disorders, and so that's what we're really focused on now. And I'm going to review some of the overview some of the data that we thought was really important in guiding those leads. In terms of how NARSAD has fit in for me, and in addition to the grants that I have had from NARSAD, my mentees have also benefited from NARSAD, especially Young Investigator Awards, and so several junior faculty and postdoctoral fellows have benefited from these. And some of the reasons NARSAD grants have been so important is because to really get research that's impactful done, you need to ultimately get federal funding. You can't get federal funding without pilot data. But if you can't get a grant if you don't have pilot data, how do you pay for the pilot data? How do you pay the research costs to get those pilot data? Universities now are strapped. They have relatively little to give toward young investigators pilot experiments. And so for us, especially over the last decade, the secret sauce was finding a private funding to pay for pilot data, and most of the time that was BBRF NARSAD grants. And then, as Dr. Bornstein mentioned earlier, once you have the pilot data, then you can get your federal grants. And so, as he mentioned, NARSAD leveraged its pilot data generating grants to generate more than $10 for every $1 in terms of federal funding. And so we were able to get the major R01 grants and other major grants from federal government because we started with NARSAD grants to get the pilot data. With brain imaging, and I'll specifically talk about two types of imaging. One is PET, or positron emission tomography. And then I'll talk about MRI imaging data, magnetic resonance imaging. With PET, we've used it to look at physiology, and this is an example of a study where we looked at glucose metabolism. And then we've used MRI both to measure the gray matter volume, the structural volume of the brain, but also now you can use functional MRI to actually map brain function as well. And you can learn about different things about bipolar disorder using both types of technologies. Unfortunately, so far, we haven't found any abnormalities in brain imaging that are where the imaging device gives us enough sensitivity and specificity to use that as a validated diagnostic tool. But they've been highly informative as research tools. Just by way of example, so this is from a study that we started out with PET measures of glucose metabolism. And then when you analyze these data, one of the most commonly used approaches is to put all the brains into a common stereotaxic array that then lets you essentially average across one group and then compare it to a normative group. In this way, you can highlight where are the biggest abnormalities, the greatest abnormalities between groups. And so in this was an experiment that helped get my career started, where we were able to pinpoint a reduction in metabolism in the ventral anterior cingulate. I worked with an anatomist, Joel Price, through the years, who I relied on to help me understand the circuitry and also to decide what regions we were looking at. And he picked the name subgenual because this is in the diagram is your corpus callosum, a white matter structure. And this anterior part of it is the genu, or knee, of the corpus callosum. And what we found, this was just beneath the genu, and so he called it subgenual, and the term has caught on in the literature as other people have studied the same region. Anyway, we found that there was a reduction in both bipolar disorder in the depressed phase and unipolar disorder, and these were familial unipolar depressives. Interestingly, in the manic phase, activity was elevated. We got a clue about why the metabolism might be decreased, because in the same region, we learned that the volume was decreased, and we had the hint to look for the volume change, because when we treated the depressed patients with unipolar depression, instead of normalizing in metabolism, actually the metabolism fell a little further. So that caused us to want to see whether the volume might be different. Sure enough, it was. In both the unipolar depressives and also bipolar depressives, you saw a reduction in volume, in the actual gray matter volume. We had an unusual opportunity where a half a dozen cases, to study them for PET, everybody was unmedicated. But we had a half a dozen people who had recently been taking lithium or Depakote, and Husseini Manji, by this time, our friendship had just started. And Husseini had started to study lithium and divalproex effects and learned that they had prominent neurotropic and neuroprotective effects in experimental animals. And so we pulled out these 6 individuals and found that if you had been recently on those drugs, well, your volume was pretty close to normal. And then subsequently, Husseini and I collaborated on an experiment when he was still at Wayne State, where we longitudinally treated patients with lithium and showed that you'd increase the volume of this region. Now, what we and others learned about these volumetric differences were that they were left lateralized, and so that highlighted the importance of the circuitry, because we've learned that a lot of the abnormalities are left lateralized, so we don't understand exactly why. Also, they were decreased in familial cases, and one of the groups that had both geneticists and imagers working together found that the lower volume was associated with an increasing genetic risk. The more affected relatives you had, the smaller the volume was. Also, we learned that it's evident early in the illness course. Your very first episode, it's already decreased, but then it decreases further over time. And there's even some hint of reduction before, that's statistically significant, before you have your very first episode, if you look at people at high familial risk. Also, there was a severity relationship. If you had psychotic depression, it was more severe and actually dropped most quickly over time. And if you had chronic or intermittent illness, you also saw the more severe illness. Now, Joel Price was also a neuropathologist, and so we then obtained brain tissue from individuals who'd had bipolar disorder or unipolar depression, and then had died of suicide or of other causes. And we studied the brain tissue in the same region now that we had the imaging to pinpoint where to look. And we confirmed that this reduction in gray matter, but we also showed that the histology, there was a reduction in glial cells, and subsequently we learned this was primarily oligodendrocytes. There are many types of glia. I'll talk about a different type of glia at the end of my talk. And the neural density was increased, and now what's known is this region, but also in other regions like the hippocampus, where you have volume decreases in these illnesses, you actually lose the dendritic trees that form most of the volume of gray matter. And also I'll show some shriveled dendritic trees that have lost synapses, and that's what they look like under the microscope. This was one of the studies that we did to try to understand the significance, and this was a group of depressed patients that had been chronically or highly recurrently depressed. And we compared them to a group of individuals who had discrete episodes of severe depression, but were able to stay well off medicines for up to years between episodes. And the difference, although there were some similarities, both of them had a reduction in hippocampal volume. Nevertheless, the subgenual anterior cingulate region was where they most differed in volume. And so they had the chronically or intermittently ill group had a lower volume there. And they also had reductions in these anterior temporal regions. And this was noteworthy because these were the regions that were most heavily connected with another structure where we were finding glucose metabolism abnormalities, the amygdala. There you had increased glucose metabolism, especially under stressed conditions in mood disorders. And here you can see some of Joel's anatomical studies from a monkey, where the amygdala projections are shown with their greatest density here. So the regions that are in black, so the subgenual region would have been here if you translate from a human to a monkey. And then these anterior temporal regions that I showed, also showing reduced volume, were also have heavy connections with the amygdala. And so Joel and I used the imaging data, and we studied several of these other regions as well. But we also had a plethora of literature from lesions that had been associated with mood disorders to help guide us. And then interestingly, there had been some surgical studies of identifying surgeries that would affect treatment refractory depression, improve it, that helped guide us. And so we were able to figure out circuit wiring diagrams, essentially, for mood disorders that involved the subgenual region was part of a broader medial prefrontal network that played a crucial role in emotional behavior of a variety of types, and also in the feeling of what happens, the visceral feeling, the control of endocrine and autonomic and even inflammatory responses to stressors or threats. And so these structures formed a circuit, and the glucose metabolism data suggests that there was reverberatory activity going on in these regions that was specific to these circuits. And the regions where you had a literature showing surgeries could actually interrupt these circuits and actually give you relief from treatment refractory depression included anterior cingulotomy and prelimbic leucectomy, which severed some of these projections between the amygdala and the medial prefrontal cortex. And these areas were tested with deep brain stimulation by Helen Mayberg, who's one of the previous prize winners, using deep brain stimulation. And they did find individual cases that would do quite well with this, even though larger trials now haven't shown enough efficacy for companies to pick this up. But this is early in the neuromodulation history, and I suspect over time they'll learn how to adapt, how to maybe change location, maybe to change frequency, where eventually these neuromodulation approaches may be quite helpful. In the United Kingdom, they use a subcaudate tractotomy that would act on just a different part of the circuit, but would implicate other areas. And similarly, there was a group studying deep brain stimulation there. Again, didn't have sufficient efficacy for a company to pick it up. And then finally, there's one of the deep brain sites, stimulation sites that's still ongoing, involves a site where anterior capsulotomy benefited depression, and so they're impacting this region where the circuit's coming between the thalamus and the medial prefrontal cortex. And then Fritz will talk about a structure that he and I studied together. So it was particularly nice to share this prize with Fritz Hinn, because Fritz and I had collaborated on a series of studies looking at the role of an epithalamic structure called the habenula. And now there's our tests even here at Mount Sinai looking at deep brain stimulation there that Fritz will mention. Eric Nestler is another Mount Sinai investigator who's been contributing in important ways to understanding what's going on in this region. And Eric, on the left half of this panel, Eric took brain tissue from people who had depression and then had died, and so he studied one group that's unmedicated, those are the bars in black, and another group that was medicated. And what he showed then was that the early gene expression was decreased in these regions, and it was the same subgenual 24 region that we had studied with our MRI and postmortem studies from our studies. And then Eric has developed an animal model that seems to simulate many different aspects of the depressive syndrome. And one of them was also this reduction in early immediate gene activation. So he could show that the animals that were susceptible to developing depressive symptoms and other changes in endocrine and behavioral that mimicked those in depression those animals had the same pattern of reduced early gene activation in the same region. He also showed that if you stimulated that region with use a new technique called optogenetics, you could actually exert a rapid antidepressant effect. And this became relevant because it's one of the new treatments that seems to have rapid antidepressant effects in both unipolar and bipolar depressives, although so far it's just been widely used at research centers, is called ketamine. Ketamine was an anesthetic drug used especially for pediatric anesthesia. And if you used sub-anesthetic levels, where it was relatively selective as an NMDA receptor antagonist, and at these levels, you could, John Crystal at Yale and others, and Dennis Charney, showed that you could get a rapid antidepressant effect. And this is a study that Husseini, Dennis, and Carlos Zarate collaborated on when we were at NIMH together. And they showed that you could get a rapid antidepressant effect within 2 to 4 hours that then maximized after a day and then would persist for several days. And then subsequently, it's been shown that you can repeat ketamine doses every few days and keep an antidepressant response up. And in trying to understand how was ketamine working, there's been a lot of investigators looking into this, and there are actually many different ideas about that. But one of them was particularly relevant to what we were finding with our imaging, and that was the data from Ron Duman and George Agajanian at Yale, where they took a different type of chronic unpredictable stress model of depression. And they could show that in the medial prefrontal cortex, in the rodents, that would be about the homologous region of the subgenual region. They could actually, with this stress, they could show a loss of the dendritic spine. So here you're losing synapses off of this dendritic, this dendrite, and here you see the distal tuft losing the density of synaptic spines. And then similarly, they showed that you would lose the electrophysiological responses to serotonin and other neurotransmitters and during the depressed phase. And here you see it going down to serotonin. And then in both cases, the spine density and then the electrophysiological responses all end up going back to normal after a single dose of ketamine. And along with it, so do you get changes in their animal behavior as well. Now, Husseini had been studying the effects of lithium and divalproex for many years and showed in a number of experimental animal models that these drugs would also, if you took chronic stress or repeated stress animal models, and you take this loss of spines, you could normalize those with lithium. And so the concept came that drugs that would enhance cellular resilience would ultimately restore these dendritic trees and be beneficial, just like lithium and divalproex. And now several groups have shown that if you look at like hippocampal volumes in bipolar patients, before lithium versus after lithium, you can show the volume going back toward normal. This is a study where Husseini and I gave both bipolar depressives and controls lithium. And then we could show that the bipolars would show an increase in just a couple of weeks in volume, whereas the healthy controls, there was not a significant difference. So it was unique to an effect in these individuals. In the areas that Helen was stimulating with deep brain stimulation, one of their studies, they took they put electrodes in and they studied the responses to different kinds of emotional stimuli. And they found that this subgenual region would show a bias toward responding to negative stimuli. And they didn't have any healthy individuals, so they didn't know whether this was something that was abnormal in these depressed patients or not. But in a variety of types of neuroimaging studies and also behavioral studies, there's been a negative emotion processing bias identified. And this ventral anterior cingulate region is one of those regions that shows a negative processing bias. And so this is one of my mentees, Teresa Victor, and this was a study on which she did her doctoral thesis with Arnie Ehrman and I as her co-mentors. Arnie had developed a backward masking test that let you show either sad or happy faces below the level of conscious awareness, and then they were only showed briefly for 26 milliseconds, and then you'd immediately replace them with a neutral face. And individuals who went through this weren't consciously aware they'd seen the sad or happy face. And what we found was that the amygdala, hippocampus, the structures I've been talking about, mediated this bias. But the region that showed the biggest effect post-treatment was this ventral anterior cingulate region, where you had an opposite pattern compared to healthy controls, and also pre and post-treatment. So after treatment, you'd see a switch in their responses to the sad versus happy, and it was composed that the initial response to sad faces, like what was shown in those data from the deep brain electrodes, where this ventral anterior cingulate has an exaggerated response or a bias toward responding to sad stimuli. You saw the same thing in the pre-treatment phase, but then it went back to normal in the post-treatment phase, and these were individuals who did respond. And in contrast, the missing response to happy faces suddenly appeared, and they started to respond instead to happy faces in this region. And it turns out this is the normative pattern. In healthy controls, this region does not respond to the sad and instead responds to the happy. So you're restoring a normative pattern of processing bias. I just want to finish by saying another implication that we're learning about. Immunology was mentioned earlier, and you'll hear about that through the day. It looks like the innate immune system plays a significant role in the pathophysiology of at least some people with mood disorders now. And so this has been increasingly studied. And one of the ways that it's been studied is with an inflammatory challenge. It was learned that if you, in people who got interferon for hepatitis or other illnesses, about 40% would develop a major depressive episode, even though they hadn't experienced that before. And this would pick up. The longer you were exposed, the more you would get this. And so people started to study immune challenge in the context of mood disorders. These were actually just healthy individuals who were given an immune challenge with a typhoid vaccine. And they showed that you would increase a cytokine, one of the hormones of the immune system, if you will. But the degree to which these individuals felt a negative mood was correlated with the amount of activity right here in this subgenual anterior cingulate region. And this region also turned out to be the region where you had the most prominent difference between between bipolar depressives and unipolar depressives who had died by suicide. When they looked at the quinolinic positive microglial cells. I'll talk about what quinolinic acid means later, but it means these microglial cells were not in the classic inflamed state but they were in an intermediate state where they're releasing too many cytokines. Jonathan Savitz, one of my mentees who's a current NARSAD investigator, is studying these, and he's showing that an inflammation of flipping things down this quinolinic acid pathway toward quinolinic acid also influences the volumes of structures like the hippocampus in this study, and so is playing some role. So in summary, we study mood disorders based upon behaviors and emotional states. We learn from imaging about the systems that are important. Increasingly, we're learning about the cells that make a difference in bipolar disorder. And then we're now learning that proteins, including those in the immune system, are playing roles at mood disorders. And then ultimately, we'll learn more, just as you've heard about in schizophrenia, we'll learn more about how the genes are playing a role as well. And just to finish, I've tried to mention my collaborators as we went along, but I'll just leave this up for the question period.